Maturation promoting factor is a type of CDK, and it is highly involved in the control of mitosis and plays a big role in the G2 checkpoint. Now, maturation promoting factor, as we said, is a CDK. Remember that K stands for kinase, cyclin-dependent kinase. So these proteins are kinases, and we know that kinases phosphorylate their substrate or add a phosphate group to their substrate. Now remember that phosphorylation is highly involved in signal transduction, right? Those phosphorylation cascades can carry or can transduce signals in cells. So maturation promoting factor phosphorylates a bunch of things during G2 that leads the cell to crossing that G2 checkpoint into mitosis. Now, MPF is actually turned off during anaphase, and it's turned off by a negative feedback mechanism. And as we can see in this graph, it's basically the same graph I showed you before, but instead of CDK activity, here I'm showing MPF activity, so a specific CDK's activity in relation to the fluctuation of cyclin concentrations in the cell. Now, there are two checkpoints during mitosis actually. The first checks to make sure that all those spindle apparatus attachments are correct, right? We want to make sure that each cell is going to get the right number of chromosomes and that those chromosomes will get split apart, right? So this checkpoint more or less is assessing whether or not each pair of sister chromatids has a spindle attachment to it and making sure that the kinetochores have spindle attachments from opposite poles. So it wants to make sure that these sister chromatids separate and move to the right pole. So it's also checking to make sure that one sister chromatid, for example, one pair of sister chromatids doesn't have microtubules attached to it from the same pole, for example. Now the second checkpoint in M phase, or mitosis, involves MPF again. And this checkpoint controls the passage from M into G1, or mitosis, back into interphase. All right, let's flip the page.